Hello and welcome to another episode of Momento Malum today. Perennial plants are subject to a variety of stresses throughout the year. The genetic variation within a species, and in some cases their close relatives, can provide the means to create new cultivars better adapted to the environmental conditions of a particular environment. As of right now, the progress in understanding how woody plants and perennials respond to low temperatures and disease has been relatively slow and limited. To speed up this process, the Lithuanian Institute of Horticulture would use different methods to evaluate the resistance of local apple, pear, cherry, currant, and strawberry cultivars to different diseases and cold stresses, and here are the results. The first project was on a study of resistance to mildew, septoria, and anthracnose in hybrid lines of currant. Common cultivars in Lithuania of black currant were crossbred with red currant cultivars and gooseberry cultivars. Following this, the resulting hybrids were allowed to undergo open pollination and produce seedlings. Both generations of seedlings would be evaluated for disease resistance, utilizing a 0 to 5 scale, 0 meaning complete immunity, and 5 meaning 75% or more of the plant has been damaged. None of the resulting seedlings of either generation showed complete immunity. The initial F1 hybrids of red currant and black currant and their progeny showed a range of 0.58 to 1.6 on the resistance scale to septoria. These would be highly to moderately resistant in nature. The same results were found for anthracnose, and for mildew, the range for disease resistance was between 0.11 and 1.6. The gooseberry hybrids were found to be highly resistant to mildew, and moderately so to anthracnose. Within these hybrids, the disease resistance seems to be mostly maternally inherited. This may indicate that this trait may be at least partially inherited through the chloroplast and/or the mitochondria. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. The next experiment involved the crossbreeding of local cultivars of apple with the scab-resistant cultivar known as Prima. This cultivar is a carrier of the VF gene, which provides complete immunity to many strains of apple scab. The experiment was done to see how heritable this gene is. They used genetic markers to determine the presence or absence of this gene in 16 seedlings. Of the 16 seedlings, all were found to have this gene, indicating that this gene is highly heritable. The same experiment proved that this screening method was also highly reliable. The fourth experiment was studying the COR47 gene. This gene is used in the creation of cold hardiness in sweet and sour cherries as well as strawberries. The goal was to determine how variable the expression of this gene is, and it was revealed that the activation of this gene was most expressed after an acclimation period between 30 to 35 days. This happened both in cherries and strawberries. As a result, it was found that the maximum cold hardness of a cultivar can be determined after this period has passed. There were other experiments, but they were best described through the charts laid out in the paper. These two will be seen on the last two slides of this video. And that about covers everything. Thank you for watching.